Okay, so chemical test for biological molecules. When we are talking about biological molecules, we will study them one by one. So now, let's talk about testing for starch. Okay, so when, we are talking about testing for starch, what comes to our mind? Okay, we get iodine solution. Okay, yeah, this is also one more thing in the exam. Students usually they say iodine, but what you should say, so the reagent that you are going to use for starch is iodine solution. And what will be the result for that? So results, when we say results, positive result is going to be blue black, and negative will be that it remains orange or yellow or orange or light brown. Okay, yeah, so this is going to be the negative test for starch. Okay, one more thing, and they will ask us about the safety precaution for each test. So we should know the safety precaution when we are talking about for this test particularly is eye protection. Okay, so when we are talking about eye protection, we can talk about safety goggles. So here, safety goggles. Yes, okay. So after testing for starch, we will go for next carbohydrates. Okay, so keeping in mind this both testing for starch and reducing sugar, they both are basically carbohydrates. Okay, so we are talking about all three main nutrient molecules that we have: carbohydrates, proteins, and fats. So here in carbohydrates, we have this. Now, let me put up here. The second test for carbohydrates is test for reducing sugar. So when, we are talking about tests for reducing sugar, this is the question which actually requires more detail in the exam. Yes, this test actually, has a bit more detail as compared to the other three tests. Why? Because here, heating is also required. And how you are going to start with, you are going to talk about preparing a sample. So prepare a sample, what does it mean? Let's say if they have given you a sample that needs to be crushed, okay, that needs to be grinded. So you are going to mention that. Prepare a sample, how? By grinding, okay, grinding, or you can say crushing, even cutting, okay, or let's say if it is the liquid one. So, you may dissolve it. Okay, it is liquid, or it is like, in that powder form or something that can be dissolved. So you can dissolve it in water. Means you may make a solution. So this will be your first step where you are going to prepare a sample. After that, what are you going to do? You are going to put Benedict solution in it. Benedict solution. Then what is the important point of this test is heat it. Yes, you have to heat it using what? Using water bath. So when you mention this water bath means you are actually talking about the safety precaution as well along the way. Okay, so yeah, after that, heating it using the water bath, now you will observe the change. So when you observe the color change, what you will see here also, there is one more important thing when you are observing the color change in results. Okay, it will change from what? From blue to. This is very important in the exam to mention about it. Okay, not just simply that it will change into red, orange, or what. It will change from blue to what? Okay, so yeah, here, we will mention blue to what? Blue to green, orange, or red depends upon the quantity. Okay, so the more sugar is present, the darker the color is going to be. Yeah, so this is our test for reducing sugar, which actually has most of the steps and the safety precaution also, particular safety precaution, which is about using water bath. Other than that, you may use general safety precautions like, they have using about using the safety precaution using eye protection, right? Yeah, about lab coat. So those are general ones, but this is something, which will be used particularly for that. Okay, so we have already discussed about testing for starch, testing for reducing sugar. Now let's move on to testing for protein. Yes. So, okay. So now testing for protein, when we are talking about, okay, so as I told you earlier that preparing a sample, it is a general step, okay, which may be used for all of these tests or some of these tests. It really depends upon the sample that they have given you, okay. Yeah, so the main thing that you have to remember is which reagent that you are using here. So here for testing for protein, what are you going to use? You are going to use Biuret solution. What I'm saying, Biuret. Biuret solution. Okay, this Biuret solution. This is basically the combination of copper sulfate and copper sulfate, CuSO4, and NaOH, sodium hydroxide solution. What will be the result if proteins are present there? So the result, when we are talking about the positive result will give us that positive will be, it will change from blue to purple. And if it is negative, what will happen? It will remains blue. In the exam, this is very important to talk about that, which color to which color it is going to be changed. Like here, as I said, for positive result, it will be blue to purple. Okay, you may say purple, violet, lilac, yeah, but do mention about it. Otherwise, it will remain this. Also keeping in mind what will be the safety precaution for that, since it is a general one. So safety precaution for that you can use is eye protection, which is actually the safety goggles.
Okay.
Yes, now we will talk about our last test. Okay, we are already done about testing for starch, testing for reducing sugar, testing for proteins, and yes, now we have the final one, which is actually testing for fats. Yeah, this is very interesting, and here I'll draw a diagram for you as well to make you understand. So here, we are talking about testing for fats. So keeping in mind, what do we call this test? This is basically called ethanol test. Okay, or a particular word is used for that. What do we call it? We call it emulsification. Okay, emulsification. What is that? So testing for fats, what are we going to do? Of course, we are going to take a sample first. Okay, take a sample where? Take a sample in the test tube, in one test tube, and dissolve it with ethanol. What does it mean, dissolve it with ethanol? We already know that ethanol is what? It is a better solvent, right? I write here. So here I'm taking one of the test tube. Here, I have the sample and ethanol, and now what? I will dissolve it and it will be dissolved. So here, what do I have? This is my sample that they gave me, plus ethanol. because ethanol is a better solvent, keeping in mind, like, let's say when, you have, let's say, for example, if you have oil, okay, it will not be dissolved in water. You will still be able to see the droplets, right? Since, it doesn't dissolve. But if you put it in ethanol, it is actually going to be dissolved. Yeah, this is what we meant. Now, next step after, so here in this one test tube, we have sample plus ethanol. Now, what are we going to do next? Now we are going to take water in the other test tube. Okay, so since now we are going to take water in the other test tube, what is that? Let me draw it so, we understand better what I meant to say. So not in this test tube, huh? This is what students usually, they get confused. So I am going to take another test tube. And now, what I have done, I have put water in it. So what is that? This is actually water. Now, next, what I am going to do. My next step is put sample plus ethanol in the test tube containing water. What do I mean? Let me show you. Here, this is your sample. What are you going to do? You are going to put in this. And now, after, you put in it, what is gonna happen? You will see a white emulsion. Okay, let me show you a white emulsion. Okay, that looks cloudy or milky. That looks cloudy or milky color, I should say, or a milky color. Okay, and this is going to be a positive test. What I'm saying, that looks, I should use a different color. Okay. This one. So here, once you put it here, you will see this white emulsion. Okay, since, it does not dissolve, right? So here, what will be, this will be your white emulsion. This is what, I am referring to. This will be your white emulsion. And what does it mean now here? Let's conclude it. Okay, so this is going to be the positive test for fats. Means formation of this white emulsion is the positive result, positive test for fats. And let's say if it does not happen, okay, if it does not happen, let me complete it. If it does not happen, what does it mean? The extract, the sample, it does not contain fat, does not contain fat. Let's conclude it. What else you are supposed to know in this? Again, the safety precaution. So wherever, we are talking about, as you guys already know here, we are talking about ethanol. Okay, I am putting ethanol in it. So whenever, we talk about ethanol, whether chemistry or biology, we know that what is the safety precaution, that it is flammable. So you need to keep it away from the fire. So this is how, we have done all our 4 tests we have completed.